Yeah, just to kind of give a brief outline of the day. So we have 3 speakers before the break, Matt Kaeberlein, Brian Kennedy, and Evelyn Bischoff. And again, from Mauritius Longevity Summit perspective, these are, 3 of the most eminent speakers in the planet, literally. And they're giving an overview of what is aging research and how it is applicable. Brian is looking at clocks and how they're useful currently, and Evelyn is taking us into the clinic. As we go through the day, there'll be a little bit more of the geroscience hypothesis of aging, the biology of aging and how it affects different disease patterns, and we will look at issues around neurodegeneration and cardiovascular disease as things go on. So the day will have that sort of a narrative to it. And they're all really top-class speakers. I just wanted to speak briefly about the first 2 speakers, because again, for those that don't know, longevity as a phrase, as a kind of a movement in industry has become immensely popular in the last 5 years, 3 years. But I think speaker 1 and speaker 2, Matt and Brian, really are among the founders of this whole thing. So the work, both of these gentlemen worked in the same lab for a period of time. They'll argue as to who was there first, and if we're talking 1950s or exactly when this happened. But they were there in a similar era and did fundamental work in things like sirtuins and the mTOR pathway and so on. All of the things that I think spawned what everyone is excited about now. At the fundamental level, their work as scientists has been pivotal. And it's great to see them go from, scientists to really influential individuals now. So just to go to Matt first. Matt, as I say, is one of the founding scientists in this field. He has looked at geroscience and its translation. He was involved in a project called the Dog Aging Project, where quite a number of different interventions are tested with our favorite pet animals. He has recently opened Optispan Clinic and the associated Optispan podcast, which I would recommend all of you to look at if you want to see actual aging research being discussed in an accessible way. And yeah, without further ado, then can I introduce Dr. Matt Kaeberlein, please.
Talked a little bit about my background. I think that was a nice way of saying I'm old and I've been around a long time in this field. But for those of you who don't know me, my training really is as a basic scientist. So I spent 25 years studying the genetics, molecular biology, biochemistry of longevity. And then a couple of years ago, I left my academic position to start Optispan, which really I would frame as a healthcare technology company. Our goal is to create enabling technologies to allow medical professionals who want to practice what I'm going to call health span medicine. I already have become a little bit dissatisfied with the term longevity medicine, not because that's not what we're doing, but because that's already become associated with, I think, some things that are maybe not as rigorous or scientific as we would like them to be, or at least I would. But really we're about creating those technologies to enable scaling of this kind of care for as many people as possible. And so I tell you this because my lens is very much influenced by my training as a basic scientist. And so a lot of what I'm going to say today is going to be around what I view as the need for more precision, more rigor in the way we think and talk about this science and this type of healthcare. So I was asked to give a little bit of a foundational background on the biology of aging and geroscience, so I'll do that. I'll talk a bit about some of the common misperceptions, and then I think some of the important facts around this area of research in medicine, and then I'll tell you a little bit about how I see the overall landscape of the longevity healthcare ecosystem. So we've talked a lot about biological aging already in this conference. But I just want to make sure everybody's really on the same page of what we really mean around the concept of biological aging. So I think dogs are a really powerful way to illustrate this concept. Probably everybody is familiar with the idea that dogs, 1 human year is about 7 dog years. That's just another way of saying dogs age biologically about 7 times faster than people do. So first of all, foundational, biological aging does not equal chronological time or chronological aging. It is a biological process, which means that it is under the control of both genetic and environmental factors. And we know a fair amount now about those genetic and environmental factors. Certainly nowhere near everything, but we've learned a lot about the things that influence the rate of biological aging. As you can see by considering humans and dogs, there's variation in the rate of biological aging across species, right? Some species age faster, some species age slower. But importantly, within species, also different individuals can age in different ways and in different rates. And dogs are useful here as well. If any of you are dog people, I'm sure you can appreciate that big dogs age biologically faster than small dogs do. They have a shorter life expectancy, they develop most of the same functional declines and diseases just at an accelerated rate. Big dogs age faster than small dogs. There's actually evidence that that's probably true in humans, and it's definitely true in other mammals like mice, this body size effect. So different individuals within the same species can age at different rates biologically. A couple of points I want to make, just again for clarity purposes. Biological aging is a concept. It's qualitative. It's not quantitative. We do not have any way to measure biological age or biological aging. There's no consensus definition on what biological aging is. We don't understand it well enough. So I say that because many of us, you've probably heard it said multiple times in this meeting already. You'll probably hear it said multiple times going forward. We measured biological age. We used this clock to measure biological age. And I'm just going to say, no, you didn't. And I do this myself. I slip up as well. But again, I think if we're going to be able to communicate what we're talking about, broadly speaking, we need to be precise in our language. So I'll come back to this, but every time you hear somebody say that they've measured biological age, just think in your head, no, you didn't. In fact, if it wasn't 9 a.m. and it wasn't a longevity meeting, I'd suggest we make it a drinking game. Every time, so Brian's going to talk about clocks next. Every time he comes out, he says, I measured biological age. Everybody has to take a drink. We'd be hammered by the end of Brian's talk. So we just need to be precise in our language. And I can say that because Brian and I are really good friends and we tease each other all the time. Okay, so I'll come back and touch on that in a minute. The other thing to appreciate is the rate of biological aging is malleable. It changes, it's dynamic, right? I think we're learning that just throughout the life course, there are periods where biological aging may speed up, periods where it may slow down. If you look at dogs to humans, this idea of 1 human year is about 7 dog years. That's just an estimate over the entire lifespan, right? But really, early in life, dogs are aging biologically, we think, maybe 15 or 20 times as fast as people are. And then it sort of tails off to maybe only 2 or 3 times as fast as people are later in life. So I want you to appreciate it's not static. It doesn't stay the same. The rate of biological aging can be changed, and it probably does change even throughout our lifespans. Okay. Again, just sort of foundational background. We've heard of the hallmarks of aging, talked about a few times already. Probably many of you are familiar with the hallmarks of aging. If you're not, I'm not going to take you through the entire list. But it is important to appreciate the hallmarks of aging are really powerful. They have been a really useful tool to sort of describe what we understand about the biology of aging. So these are apparently highly evolutionarily conserved factors that are tightly associated with biological aging, in some cases probably causal, at least for the functional declines and diseases that we associate with biological age. And that's really powerful because, one, it helps us talk about it, and it also gives us targets to go after therapeutically to potentially slow the rate of aging, increase health span, increase lifespan. And they're incomplete, right? So this is a set of 12. It started at 9, 12. Now it's 3 more have been proposed. So it's a work in progress. This doesn't represent everything about biological aging. So imperfect, I think everybody in the field recognizes this. Incomplete. And the last point I want to make is they're interconnected. So if you look in the middle of that circle, there's this kind of network looking thing. The hallmarks are connected by a network of proteins, metabolites that, again, we kind of understand. Things like mTOR are key nodes in this network. Insulin IGF-1 signaling that we heard about yesterday as a key node in this network. And we've learned that if we tweak these key nodes, we can modulate all of the hallmarks of aging simultaneously. That's kind of how we know they're interconnected, apparently slowing the biological aging process. I think it's still an open question whether there is one biological aging process, and I think we still don't really know the answer to that. But we can apparently modulate that whole suite of hallmarks of aging with single genes or with single drugs to slow aging, increase health span, increase lifespan, at least in laboratory animals. And I feel confident saying we can do this in humans, but it's hard to prove. One more sort of definitional thing that I think might be useful. We've heard this term geroscience already talked about quite a bit, but nobody has really defined what is geroscience. So I think there are a few different variations on this definition, but really, for me, what geroscience encapsulates is the study of the biology that connects aging to age-related functional declines and disease, right? And that's important because in most developed countries in the world, the vast majority of chronic disease has biological aging as its single greatest risk factor. In the United States, 9 of the top 10 causes of death have biological age as the greatest risk factor. And so if we can understand those biological mechanisms, that gives us the opportunity to have an impact on multiple functional declines and diseases that are very relevant for health of the overall population. And if you look at even just the magnitude of the effect of biological age on these diseases, it's quite striking. So these are 4 of the top 10 killers in the United States in 2020 and 2021. So COVID-19 is there, but you can really think of that as, deaths due to viral infections, generally speaking, influenza and other viruses. And so these are some of the common risk factors that are talked about in the traditional healthcare system as being important for your likelihood of getting these conditions and ultimately dying from them. So things like, do you smoke? Are you obese? Do you have high blood pressure? If for some diseases, whether you're a woman or a man. The thing I want you to appreciate is the magnitude of effect of these risk factors is in the ballpark of 2-fold to 4-fold in general, sometimes a little bit less than that, but usually not bigger than that. And if we compare this magnitude of effect to the magnitude of just aging biologically 4 decades, this is the same data. The same data is still there. It's not even close. We're talking orders of magnitude higher risk from the biological aging process than from the common risk factors that we talk a lot about. And this is important because if we can actually target that biology, it gives us the opportunity to reduce the risk of all of these diseases simultaneously. And again, that's what we see in laboratory animals when we tweak that network that underlies the hallmarks. And there's every reason to believe that the same thing is going to be true in humans and in our companion animals, which I think a lot about. Okay, so that's really, I think, the core premise of geroscience is being able to accomplish this in a way that keeps people and animals healthier longer, for as long as possible. Okay, so I had the opportunity a month or so ago to give a TEDx talk on the field, and they asked me to sort of talk about some of the common myths and important facts. And so I distilled this down to 3 fictions and 3 facts. And so I'm just going to take you through those. I won't spend a ton of time on this, but again, I think it's useful to appreciate some of the common myths that are out there so that you can be equipped to think about these and maybe talk about them as we're trying to communicate this ecosystem to the general public. So fiction number 1, these are things that are not true that you may hear, is that we're close to solving aging, okay? Now that's really hard to prove that we're not close to solving, like nobody really knows the future. But if you like data and you consider yourself to be evidence-based, it's really hard to make an argument that we are close to solving aging. And there's a few ways to appreciate that. One way to appreciate it is just ask, if we look in other animals where we actually can modulate biological aging, are we continuously getting better at having a large impact on the biological aging process? Can we increase lifespan and health span by a larger extent, year over year, month over month? And when you look at the data, the answer to that is clearly no. So the largest effect on lifespan in a mammal from an intervention, and this is done in mice in the laboratory, An intervention that is implemented post-developmentally. So in other words, we're not talking about making dwarf mice, we're talking about after development starting an intervention. The largest effect on lifespan ever published is an experiment that was done almost 50 years ago by Rick Weindrich and Roy Walford. That's a extreme caloric restriction. It's about a 55 or 60% caloric restriction. And then when you get to the next best intervention, which is rapamycin, which is something I think a lot about. I've studied rapamycin in my lab. I'm relatively bullish on rapamycin as a potential geroprotective intervention in people, and we're testing rapamycin in pet dogs right now for its effect on lifespan. That's the next best, and it's about half the magnitude of effect of this almost 50-year-old experiment. And then you get down to the stuff that a lot of people are really excited about, and they're not even as good at extending lifespan in a mammal as rapamycin is, not yet. Doesn't mean it's not possible, doesn't mean if you optimized epigenetic reprogramming you couldn't do much better, but it hasn't been done yet. And so again, I think all of this just illustrates that it's really hard to say, you know, we're getting close to solving aging and we're gonna all be immortal 5 years from now, when the data just doesn't back that up. So that's one myth that is sort of prevalent out there. And I sort of think about the hallmarks of aging. I said they were incomplete. I don't know how incomplete they are, but I kind of like this analogy that maybe they're about as complete as this map of the surface of the Earth in 500 BC. This is called Hecates's map. And so in this map, you'll see there's a Libya, a sort of vaguely Europe-shaped thing, and Asia that doesn't look anything like Asia, and you sail too far and you fall off the edge of the planet, right? Maybe that's about how well we understand biological aging. Maybe we understand it better. And I don't mean this to diminish what has been accomplished, because this map was really useful in 500 BC, right? People could trade with it, they could make war with it, they could get rich with it. Just like I think the hallmarks of aging and what we understand about biological aging is really useful today. And we need to also look outside the hallmarks of aging and try to figure out what don't we understand about biological aging. And so I'd like to see a little bit more discovery science going that direction. Okay. Fiction number 2, also not true. Aging has been reversed. And again, this depends a little bit on what you mean by reverse. We can absolutely reverse specific phenotypes of aging, right? I can color my gray hair black back to my youthful color, and I've reversed a phenotype of aging, but I didn't get younger, right? You can do other things. You can start lifting weights if you've been sedentary. You'll get stronger, but you didn't get biologically younger throughout your whole body. So I think there is just imprecision in the language that we use in this field sometime, where the general public, when you say you've reversed aging, they might think that we took an old mouse and made it young again. That hasn't happened. Brian Johnson, I'm sorry to tell you, is not aging in reverse. That doesn't even pass the eye test. David Sinclair's mice did not age in reverse. Those aren't the same mice. They're not even the same genetic background. So I think we create a lot of confusion when we use sloppy language in the field. Doesn't mean it couldn't be reversed, but it hasn't happened yet. And then fiction 3, and this is going to be really important for, I think, a lot of people who want to practice clinical medicine around geroscience, is to just understand the clocks are really useful as research tools right now. They do not measure biological age. So I touched on this earlier, that we can't really measure biological age. Again, you'll hear people say this a lot. That's the drinking game. Every time somebody says, we measured biological age, you get to take a drink. So ask yourself, what do these things actually measure? And there's lots of ways you can create clocks, right? Brian's going to tell you about, I think, 3 different clocks today. The epigenetic clocks are the ones that most people are familiar with. What do they actually measure? They measure average methylation state at a subset, a relatively small number of specific positions in the genome, usually from blood, sometimes from saliva. That's actually what's being measured. Average methylation state at a small subset of sites in the genome from blood or saliva. Those patterns, each clock is a different set of methylation sites. Those patterns are correlated to something. They're correlated either to chronological age, those are generation 1 clocks, or to mortality risk, or to specific disease risk, or in a few cases, they're correlated to change in health over time. Those are the pace of aging clocks. So measure methylation state, correlated to something, and each of those things, chronological age, mortality risk, age-related disease risk, each of those is correlated to biological age, which we don't have any way to measure. So just appreciate, at best, these clocks are a correlation to biological age. They can still be useful, but they're not measuring biological age. And I think I'll touch again on the clocks and why, at least right now, I don't think these clocks, especially the ones that are available on the consumer market, should be used in clinical practice. So those are the fictions. Now let's talk about 3 facts, which are just kind of, to get the foundational ideas of geroscience. First is biological aging is modifiable. So I said it's under genetic and environmental control. If we understand how to tweak those things, we can modify the rate of biological aging. We know lots of ways to do this in mice. I already mentioned the caloric restriction experiment and rapamycin. And you can see, I was telling the truth. So you can see the caloric restriction experiment has a much larger effect on lifespan in mice than rapamycin does. So we can modify biological aging either direction. And optimally, we would want to modify it in the way that increases health span and increases lifespan. Fact number 2, targeting aging is way more effective than targeting individual diseases in isolation. And again, this concept of, currently healthcare is, in general, reactive. We typically wait until people are sick, and then we try to do something about their condition, right? Targeting aging in a proactive way has a much larger effect on health span and lifespan. There's a variety of ways, it's sort of intuitive, but there's a variety of ways you can even try to quantify this. So J.L. Shansky looked at demographic data from the Center for Disease Control in the United States on what people die from. And he asked a very simple question. If you take a typical 50-year-old woman and you assume that we can cure cancer today, all cancers, what is the impact going to be on her life expectancy, her remaining life expectancy? Turns out it's about 3 years. Same thing if you cure heart disease. If you cure both of them, it's about 6 or 7 years. If we can slow aging in people, comparable to what rapamycin does in mice, it's a much larger effect on life expectancy, with the added value that most of those years are predicted to be spent in good health because we've slowed the onset and progression of all of the age-related functional declines, or at least most of them, and diseases at the same time. So way bigger bang for the buck by slowing aging compared to reactive disease care. And then fact number 3, I said the hallmarks of aging are useful. I absolutely believe we know enough to modulate biological aging today in both companion animals and in people. And that's what I spend all of my time thinking about now. So I started as a basic scientist working in yeast, trying to find genes that affected lifespan. Now I spend all my time in the real world trying to think about how do we bring as much of this information forward, test it, implement it. And so Martin mentioned the Dog Aging Project. That's a project that I co-founded along with Daniel Promislo and Kate Creevy more than 10 years ago now, where we are both exploring biological aging in people's companion dogs. So these are pet dogs living with their owners in the United States. There are more than 50,000 Americans participating in geroscience research through the Dog Aging Project. We're trying to understand what are the most important genetic and environmental factors that influence health outcomes in companion dogs. And then there is a clinical trial that Martin also alluded to of rapamycin. This is a double-blind, placebo-controlled, randomized veterinary clinical trial of rapamycin, trying to test, does rapamycin slow aging in companion dogs? Lifespan is the primary endpoint, a bunch of health span metrics as secondary endpoints. And then Optispan, which I briefly mentioned as well, is now my day job, where we're really trying to ask, what can we do in the clinical space today to optimize health for as many people as possible? And I just want to mention, I think, and again, this is my belief, I can't prove it, I think slowing aging in companion animals is a solvable problem. In fact, I'm as confident of that as I can be of anything as a scientist. I don't know how much it'll cost, but I'm pretty confident that if we threw $100 million at this problem, we could significantly slow aging in our pets. 20%, 30%, maybe more. $100 million is a lot of money, but compared to what we spend that type of money on in society as a whole, it's really a pretty small price tag to slow aging in our companion animals. And there are some companies doing this now. So the folks at Trivium Vet, friends of mine, actually just got FDA conditional approval for a rapamycin for companion animals. And then Loyal is another company that I think probably next year will get conditional approval by the FDA for the first drug to slow aging in companion animals, which will be an important milestone for the field. So exciting stuff happening there. He coined the term P4 medicine. And I would add a 5th P to that. I think if we're going to be successful here, it's got to be pragmatic, right? This kind of approach, you can't measure everything. You can't do multi-omics on everyone. We've got to be pragmatic and understand what's affordable. What is the 80-20? Where can we get 80% of the value for 20% of the cost? Sure, some people can go to very high-end concierge clinics, and that's great. I'm all for it. We run one at Optispan. But we really want to figure out what can we bring to everybody, or at least the majority of individuals in society. And I think it's doable. I think there are opportunities to scale this, or I wouldn't be doing what I'm doing now. It's got to be scientifically rigorous. Really, that's my biggest concern in this space right now, is that there is a lot of stuff happening that isn't very scientifically rigorous. And that has the potential to really damage the credibility of this ecosystem before it really gets off the ground. And as part of that, whatever we're doing in these clinics has to be medically and ethically sound. So I've, again, as I said, learned a lot, and I've got some ideas around what I've learned from building this at Optispan works today, and a couple of things that don't really work in the clinic today. We focus, of course, on the foundation. Lifestyle really, for most people, is going to be the biggest lever you can pull when you get a lot of information about them to improve their health span. We talk about lifestyle through 4 pillars: eat, move, sleep, connect. We like active words. It means what you think it means. Eat is nutrition, move is exercise, sleep is sleep. Connect incorporates both sort of personal sense of purpose, mindfulness, and the importance of human relationships. I'm certainly not going to argue one is more important than the other. I think it's very individual where people can get the biggest impact on their health going forward. Like I mentioned, it's important to avoid the noise that's out there. We use a lot of proactive diagnostics and biomarkers, and I'll just show you a few of those, and you've heard about some of these already. And we use proactive approaches with therapeutics, evidence-based proactive approaches, personalized, particularly with already approved drugs. I think there's a lot of opportunities there, some of which we have reason to believe could be geroprotective in that they modulate the biology of aging. And I think it's important, I'm going to come back to lifestyle for a second, because I think it's important to appreciate the reason why these lifestyle pillars have the largest impact is because they modulate the biology of aging. The reason why when you eat a healthy diet and you sleep well and you've got good relationships and you exercise, you are less likely to develop dementia and cardiovascular disease and diabetes and every other age-related disease is because these things modulate the biology of aging. They are true geroscience therapeutics, and I think it's just important to communicate that to people. Okay, so this is sort of a brief overview of some of the things we use that we think are really valuable types of information to get from people. Comprehensive blood work, that makes sense. And I think a lot of people are doing this, slight variations, but if you look at what people are doing for comprehensive blood work in the health span medicine, longevity medicine space, there's a pretty consistent overlapping set. We think about this through the lens of systems of health, and we try to take all the different types of biomarkers you can look at and understand how the different systems are functioning. Imaging, I think DEXA is something that should just be part of everybody's foundation, probably annually once you get into your 40s and 50s. Super powerful tool, and gives a lot of information, not just about bone density, about muscle mass, about metabolism, where is the fat located. We use a variety of other types of imaging tools. We do advanced cancer screening. That I would put in the sort of second tier. That's not right for everybody. Here I'm talking about things like, you know, the Gallery test, the blood-based test that can detect 20, 30, 40 different types of cancers. Genetics is really important, understanding your genetic risk. We do a little bit with wearables. Those are going to become more and more powerful. Oura Ring, Apple Watch, things like that. Lifestyle assessments to understand where people are at in their health span journey, so we can identify which levers are going to give us the biggest pull. And then we create a personalized health optimization plan with a combined team approach of both the physician and a coach. And there's a lot of power there if you get the right team in place. And so this is working. I mean, again, it's not going to shock you to hear really powerful success stories when you do this sort of proactive care for people and you can get them engaged. So I just want to briefly touch on some things that we started using and we stopped because they just don't work in clinical practice. And these are the sort of broad microbiome tests, especially again, the ones that are on the direct-to-consumer market.
and in particular, the biological age test. So I alluded to this earlier. So why would I say don't use these tests? It's pretty easy, actually. We have no information on precision or accuracy. We have no idea how close these tests get to the true value. That's a real problem in a clinical diagnostic. They have not been validated for clinical use. And if you don't know how close they are to the true value, you have no way to know if you see a change over time, if that's real or not. So that alone should be enough to suggest that maybe we shouldn't be using these in clinics right now. And part of the reason we have no idea what the precision or accuracy is there's no industry quality control, right? These companies are just doing whatever they want. There's no regulatory validation. And then in general, these aging clocks are not actionable, particularly the epigenetic clocks. You get a number back, but it doesn't tell you what to do. Brian, I think, is going to tell you in a minute about a clock that's based on blood chemistry markers that we understand. That is actionable. There's actually a lot of power there. So I'm not suggesting these clocks have no value, but particularly the direct-to-consumer epigenetic clocks, I would just stay far away from right now in clinical practice. Okay. And then I'm almost done. I'll finish up. There's a whole lot of gray area in longevity science today. And this, I think, is where a lot of the potential is and a lot of the risk is. And I think as a field, we just need to be careful to think about how we navigate the gray area. Supplements are really problematic. We have no idea what works and what doesn't work. That's a challenge. And so our philosophy is to be conservative on supplements. We recommend supplements in a very specific way, particularly things that we can measure and we can supplement people into the optimal range. But by and large, I think there is too much use of supplements in this space that have very little evidence to support them. Then there's a bunch of stuff. Peptides are super interesting. Some of them are very useful, very powerful. We don't have a ton of data on what works and what doesn't work, and some of them are dangerous. It's just a fact. But a lot of people are out there using and prescribing peptides in what I would consider a pretty reckless way in the absence of evidence for safety and efficacy. And then there's a whole bunch of wellness stuff that, again, may or may not have real value, but are popular, and we just need to be thoughtful about how we talk about these things in these longevity clinics. Therapeutic plasma exchange is a really interesting example. So that's an FDA-approved procedure. There's some evidence that it can actually have benefits. I've done it myself. I didn't feel anything other than I was really tired that evening. But probably not ready to be rolling out, broadly speaking, to the general population, right, for a variety of reasons. And then gene therapy, I think, is like I think Brian said this yesterday. There's no reason people should be going and getting gene therapy for longevity. We don't even have any idea what genes to modulate for longevity in humans. So to me, that is getting way ahead of the curve in terms of what the data actually supports. I'm sorry if there are any of you who run gene therapy clinics, but that's my pretty strong opinion there. Okay. So how do we improve? Better science and data. So it would be great if we could figure out a way to aggregate data from all of these clinics, all of these providers that are using experimental therapies, and have confidence in that data. We don't have that right now, which is why we don't know. Do stem cells work? Which stem cells work? Do exosomes work? We don't know because nobody's collecting the data in a way that we can trust. What do the side effects look like? We don't know. Do you really think the clinics that are doing these things are going to be honest about the side effects? I don't. So it'd be great if we could do that. I think we're getting more large-scale human data sets, longitudinal, multi-omic data sets that are quite powerful, especially with the new AI tools that are coming online. And we're starting to think about clinically validated aging clocks, which will be useful in the clinic. Again, Brian will tell you a little bit about Lineage 2, which is my personal favorite right now. Hopefully, we can get to the point where we have consensus best practices in the field for at least medical providers who want to do best-in-class, evidence-based health span medicine, have some guidelines, right? Not that everybody has to do the same thing, but have some guidelines so people can have confidence in what is really backed up by the science. And then the last point I'll leave you with is I'm actually pretty optimistic. I think a lot of times, when I first started talking about how do we scale this to billions of people around the world, it seemed pretty naive. Like, I think a lot of people have the idea you'll never be able to do this sort of healthcare for the majority of people. I actually think the AI tools give us that opportunity. Even today, the latest version of ChatGPT, if you take your your blood work, your DEXA, whatever you're measuring, and you put it into ChatGPT and you ask the right questions, is really good. Is it perfect? No. But are humans perfect? No. So I think we're going very soon to get to a point where there will be these AI tools that anybody in principle could get access to and use to give them very high quality medical advice. And we're going to have to get comfortable with the idea that AI can do that. And so how that gets regulated, what that looks like, in my opinion, the way it will work best is if we have a generation of docs that are trained to use these tools and enable sort of those docs to treat a much larger number of people. But I think for the first time, I really see a path to where it doesn't have to be tens of thousands of dollars for somebody to get high quality health span medicine. And I think the AI tools will help us get there. So I'll stop there. Thank you for your attention. Martin did mention, and so I'm going to do a shameless plug, we have a podcast. It's called the Optispan podcast. I would love it if you check it out. And thanks for your attention.
A little bit short on time, so we just do one quick question.
Sure.
Anyone that is looking to be a part of the dog trial, is there any way for them to look to get into that now?
Yeah.
Anyone that's interested in their pet?
Yeah, so the Dog Aging Project website is dogagingproject.org. So unfortunately right now this is a United States only project. This is one of the things I would really like to change, but of course it's all dependent on resources. So if you have a dog and that dog lives in the United States, any dog can participate in the Dog Aging Project. Go to dogagingproject.org, nominate your dog, and then you just complete a pretty extensive survey, and then you're part of the Dog Aging Project pack.